Today we're going to talk about genes and genetic diseases and how it relates to the advanced practice nurse. When we look at causes of disease, they can either be extrinsic or intrinsic. Causes of disease such as infectious agents, trauma, toxicity, radiation, and nutritional problems are all considered to be extrinsic. Intrinsic factors that are considered in the development of disease are age, sex, and genetic makeup. In looking at the development of disease, it is always important to look at hereditary as well as environment. The objectives of today's lesson are to describe the role of DNA, RNA, and the proteins in human life, explain the role of chromosomes in maintaining human life, define mutation and its role in change of human life and disease, discuss the element of formal genetics, phenotype, genotype, dominance, and recessiveness, trace common diseases according to their mode of inheritance, autosomal dominant, autosomal recessive, and X-linked. Explain the terms penetrance and expressivity and provide an example of each, and review the role of family history in caring for clients with or at risk for genetic diseases. After the successful completion of the Human Genetic Genome Project, HGP, in 2003, significant advances have taken place in the field of genetics and genomics. One of the most important uses of genomic information is the movement towards personalized medicine. Personalized medicine is defined as the emerging practice of healthcare that uses an individual's genetic profile to guide decisions regarding prevention, diagnosis, and treatment of disease. According to the National Institutes of Health, the immediate impact on healthcare resulted in improved diagnostics, more targeted treatment strategies, and increasingly individualized prevention options. The majority of health conditions, disease risks, and therapeutic strategies to treat conditions are results of interplay between genetics and/or genomics influenced by environmental factors. Major chronic diseases such as heart disease, cancer, stroke, and diabetes have a genetic or genomic component. This knowledge is important as we conduct health histories and assessments, gather and interpret data, and identify clients who may be at risk for genomic disease, or who are suspected of having an inherited disorder. Based upon the data collected from the client, the advanced practitioners can use this genetic genomic assessment knowledge to provide appropriate counseling, genetic testing, case management, and genetic referral. Understanding key concepts related to genetics, genomics is integral when conducting a genomic assessment. Genetics is the study of single genes and their effects on the body. Genomics is the functions and interactions of all the genes in the genome. Let's start with a review of the pathophysiology. DNA is found almost entirely within the nucleus of the cell. As the cell begins to divide, nuclear material arranges itself into strands called chromosomes. Chromosomes consist of many DNA molecules arranged in sequence. Genes are considered subunits of the chromosomes and are segments of DNA that control the development of specific traits. The sequence of nucleotides in a gene determines which proteins are found in a cell, and these proteins determine both the form and the function of a cell. The sequence of the nucleotides in DNA are in a linear sequence. Transcription is the transfer of genetic information from DNA to messenger RNA. Translation uses this information to assemble amino acids into peptides, aiding in protein formation. Let's think of it this way. DNA is the carrier of genetic information for protein synthesis, and RNA, including messenger RNA, executes the instructions contained in DNA. The analogy of DNA as a cookbook filled with protein recipes is useful. A temporary working copy of a DNA recipe, or a gene, is transcribed messenger RNA, much like the recipe a cook might write on a card to avoid messing up the master copy or the cookbook. The transcribed recipe, or messenger RNA, is a list of ingredients, amino acids encoded as a list, to be assembled in a particular order. As in cooking, the order of mixing ingredients is often critical. Ribosomes and tRNA are like the countertop or mixing bowl and the measuring scoops, reusable tools needed to assemble the ingredients properly. Like cake recipes in which the batter is prepared according to one recipe and the frosting according to a different recipe, several different polypeptides may be synthesized independently, then later put together as a single quaternary protein. This is analogous to several layers of cake and frosting later being put together to form a single cake. Haploid means single. Each haploid cell has a total of 23 chromosomes. Haploid cells are formed from diploid cells by meiosis. Meiosis is the two stages of separating to create two gametes that can then combine to have both parent traits within the new zygote. Somatic cells are all other cells in the body. Diploid replication is defined as the copying of DNA to form identical daughter molecules. Transcription is defined as the message encoded within the DNA transcribes into RNA and is carried to the ribosomes. Translation is the genetic message is decoded and converted into the 20-letter alphabet of the protein structure. Phenotype. An example of this would be an infant born with the genotype for PKU. If left untreated, this can progress into mental retardation, which is a part of the PKU phenotype. This would be as a result of the environment in which the infant did not receive care. The definition of genotype and phenotype are often confusing for students, especially the idea that the environment influences the phenotype. The proportion of genotypes that actually show expected phenotypes is called penetrance. The frequency under given environmental conditions with which a specific genotype is expressed by those individuals that possess it is usually given as a percentage. The composition of genes at any given locus is known as the genotype. The outward appearance of an individual, which is the result of both genotype and environment, is the phenotype. For example, an infant who is born with an inability to metabolize the amino acid phenylalanine has the single gene disorder known as phenylketonuria, or PKU, and thus has the PKU genotype. If the condition is left untreated, abnormal metabolites of phenylalanine will begin to accumulate in the infant's brain, and irreversible mental retardation will occur. Mental retardation is thus one aspect of the PKU phenotype. By imposing dietary restrictions to exclude foods that contain phenylalanine, retardation can be prevented. Although the child still has the PKU genotype, a modification of the environment, in this case the child's diet, produces outwardly normal phenotype. When a trait is dominant, it is indicated by a capital letter. Recessive traits are shown as lowercase letters. So there are three possible genotypes: capital A, capital A, lowercase a, and lowercase aa. Having two of the same genes, whether capital AA, which would be dominant, or lowercase aa, which would be recessive, they are known as homozygous. If the individual has a capital A and a small a, which would be a dominant and recessive, then they are known as heterozygous. A dominant trait is always expressed, while a recessive trait has to have a double dose to be observable. A pedigree chart summarizes family relationships and shows which members of a family are affected by a genetic disease. Inherent patterns in families of conditions due to faulty genes. Let's first define autosomal dominant, or AD. It's defined as a gene on one of the autosomes that may be expressed even if only one copy is present. The characteristics: 50% chance of parental transmission. Both males and females are affected. The phenotype is observed in multiple or every generation, with each affected person having an affected parent. There's vertical transmission on the pedigree. Autosomal recessive, or AR. It's defined as a genetic disorder that appears only in a patient who has received two copies of a gene mutation, one from each parent. Two genetic mutations on the autosome. The characteristics: 50% chance of the offspring inheriting one gene mutation, 25% chance of inheriting the disease or phenotype, horizontal transmission on the pedigree. Proband may have affected siblings, not in the parents because they're carriers, offsprings, other relatives, and males and females are affected equally. The X-linked dominant. This definition is genetic mutations located on the X chromosome. It requires only copy of the phenotype or disease in male or female. The characteristics: the disorders are rare, but most disorders are lethal in pregnancy with the male fetus. No male-to-male transmission. There is no carrier status, and affected males transmit to affected daughters only. And then we have the X-linked recessive, which is defined as genetic mutations located on the X chromosome, and it requires both X chromosomes to be affected in females. Males with affected gene will have the disorder. The characteristics: typically males are affected, it's rare in females, with transmission from the mother who is the carrier. 50% of females will inherit the gene from their father and are unaffected carriers. There's no male-to-male transmission. Huntington's disease is an example of an autosomal dominant disease. It's also known as Huntington's chorea. It's caused by degeneration in the cerebral cortex and the basal ganglia. The defect is on chromosome 4. It's progressive in nature, with onset usually evident between 25 and 55 years of age. It is characterized by uncontrollable jerky movements, that's called chorea, which literally means dance. It typically progresses to mental deterioration and ultimately death. Symptoms usually first appear in early middle age, thus those with Huntington's disease often have children before they know they have the disorder. Other examples of autosomal dominant genetic inheritance include adult polycystic kidney disease and Marfan's syndrome. This actually illustrates the pattern of dominant inheritance. Cystic fibrosis is an example of an autosomal recessive chromosome disorder. Cystic fibrosis is characterized by endocrine gland dysfunction, which manifests in multiple body symptoms. Cystic fibrosis can be lethal. It is seen in approximately 1 in 2,500 births, and 1 of every 25 Caucasians are carriers of the disease. Other examples of autosomal recessive genetic inheritance include sickle cell anemia. This is where you, as the advanced practice nurse, need to have some cultural considerations. It occurs in descendants of individuals from sub-Sahara Africa, South America, Cuba, Central America, Saudi Arabia, India, and the Mediterranean countries such as Turkey, Greece, and Italy. In the United States, it occurs most frequently in African Americans. Another example of an autosomal recessive genetic inheritance is Tay-Sachs disease. Approximately 80% of the individuals diagnosed with Tay-Sachs are of Jewish ancestry. PKU is another example, and it's most commonly found in Caucasians. And color blindness is also an example of autosomal recessive genetic inheritance. This is an illustration of the autosomal recessive gene. The X-linked genetic inheritance. Because a man transmits an X chromosome only to a daughter, there is no father-son transmission. Hemophilia A and B are examples of X-linked recessive disorders transmitted by female carriers and displayed almost exclusively in males. It's characterized by prolonged coagulation time that results in prolonged and sometimes excessive bleeding. and this illustrates the sex-linked genes. In chromosomal abnormalities, chromosomal disorders have been associated with mental retardation, failure to thrive, developmental delay, low birth weight, infertility, and histories of frequent spontaneous abortions or stillborns. Approximately 50% of spontaneous abortions have chromosomal abnormalities. An estimated 1 in 160 newborns have a chromosomal abnormality. For example, a newborn with trisomy 21 or Down syndrome has an extra chromosome 21 and therefore has a total of 47 chromosomes instead of the usual 46. So let's talk about DNA structural abnormalities. There are 4 of them: deletion, duplication, inversion, and translocation. Deletion is when genetic material is lost. In the illustration on the slide, it shows the fat cat ate the wee rat. And when deletion happens, what you see is the wee rat. Duplication is the repeated gene or gene sequence. It's actually an exaggeration of the phenotype. So as you see on the slide, the fat cat ate the wee rat turns into the fat cat ate the wee rat. Inversion is the linear arrangement of the genes on a chromosome, and it's broken, and the order of a proportion of the gene complement is reversed. You see this in hemophilia, and you can also see the illustration of the fat cat ate the wee rat example on the slide. Translocation is the transfer of a part of one chromosome to a non-homologous chromosome. This is illustrated on this slide as the fat cat ate the wee rat house upside down, and this is seen in cancer. So let's just take a look at chromosomal cell abnormalities. Aneuploidy, the cell does not contain a multiple of the 23 chromosomes. In trisomy, it's a cell with 3 copies of one chromosome. It's actually a form of aneuploidy. And monosomy is the presence of only one copy of a chromosomal pair in a diploid cell. This is lethal if it occurs in autosomes in newborns. In general, loss of chromosome material has more serious consequences than the duplication of chromosome material. Aneuploidy, there is autosomal aneuploidy, which is a trisome that can occur with any chromosome. Trisome of the 13th, the 18th, the 21st chromosomes are the most frequently seen in newborns. The trisomes of the other chromosomes are basically not survivable. Trisomy 21 is Down syndrome. It's an example of an aneuploidy in an autosome. It is seen in 1 in 800 births, and the risk increases with maternal age. In sex chromosome aneuploidy, there's 3 examples that are fairly common and generally less severe than autosomal aneuploidies. In trisomy X, instead of 2 X chromosomes, the females have 3 X chromosomes in each cell. Most of these females have no overt physical abnormality. However, They sometimes have issues with sterility, menstrual irregularity, or mental retardation. Turner syndrome is a condition that leads to somewhat more serious problems, and it is the presence of a single X chromosome and no homologous X or Y chromosome. The individual has a total of 45 chromosomes. It occurs in females. They usually have a short stature, female genitalia, webbed neck, underdeveloped neck, and imperfectly developed nipples. Klinefelter syndrome affects males. They usually exhibit small testes, some breast development, sparse body hair, and they have longer limbs. It may not be diagnosed until adolescence when the male seeks help for late puberty. These individuals have at least 2 X chromosomes and 1 Y chromosome in each cell. So why complete a family history? I'm sure by now you have started to realize why that is important. Um, and I'm sure that you are able to draw the following patterns. Autosomal dominant brown eyes when both parents have brown eyes, but 2 grandparents have blue eyes. And autosomal recessive blue eyes when both parents have brown eyes. So let's go on to multifactorial inheritance for just a minute. This is when environmental factors influence the expression of a trait. Examples of multifactorial traits include height and IQ. Although both height and IQ are determined in part by genes, they are also influenced by the environment. For example, the average height has increased by 5 to 10 centimeters over the past 100 years because of improvements in nutrition and health care. Also, IQ scores can be improved by exposing children to enriched learning environments. These examples illustrate how both genes and environment can contribute to variations in these traits. Other examples can include essential hypertension, diabetes, coronary artery disease. And this slide is just a review. The composition of genes at any given locus is known as the genotype. The outward appearance of an individual, which is the result of both genotype and environment, is the phenotype. Remember that the environment influences the phenotype. And the penetrance of a trait is the percentage of individuals with a specific genotype who also exhibit the expected phenotype. Remember the PKU example from earlier in this lecture. Now, let's go on to a case study. 6-month-old James has had frequent respiratory infections. When taking a health history, you learn that his mother and father have sisters who died at before adulthood with cystic fibrosis. Her 2 brothers were normal. How would this look on the genogram? Cystic fibrosis is an autosomal recessively inherent disease, resulted in the secretion of thick, abnormal mucus in the lungs and pancreatic insufficiency. The range of seriousness of the disease is broad, but most children require extensive medical treatment and daily multiple respiratory therapy. The disease is progressive with about 40% of cystic fibrosis patients living to the age of 30. Cystic fibrosis is the most frequently inherited genetic disorder among American children. It affects 1 of every 2,500 children. 30,000 currently have the disease. Cystic fibrosis can be caused by any one of over 100 different mutations known to occur on the CF locus on chromosome 7. 75% of all cystic fibrosis cases result from a single mutation that is easily screened. The other 25% of the cases result from other mutations, most of which cannot be tested. 1 in 20, or 12 million Americans, carry one mutation on the CF locus. That is, they are heterozygous for the CF mutation. So another thing that we're going to look at in this semester is nursing diagnosis. And one of the nursing diagnoses for this particular, for just genetic problems in general, it could relate to cystic fibrosis or any genetic problem or disease, would be anxiety related to the presence of specific risk factors. So it could be a history of a genetic problem or prenatal detection of genetic disease. Thank you for your attention, and I hope this helps broaden your health history assessment.